I think today is really my pleasure to talk about knowledge graphs. I will describe a few knowledge graph built either in my lab or built as a team. So before doing that, I would like to introduce a really big team, a really amazing team, the Punk-based team, who started 1 year ago to build a knowledge base for type 1 diabetes and islet biology. So this is really a new initiative, and we grab really amazing people to join us. Within the Punk-based project, there is a small component called a knowledge base called PanKgraph. And there's also amazing trainees in this consortium, and we are doing a really fantastic job. So I think now I will describe what is a knowledge graph. So knowledge graph essentially consists of nodes and edges. Use this way to describe a knowledge network. So here, essentially we have 5 nodes. Here we have a variant, type 1 diabetes, gene, pancreatic tissue, and ontology term. And we have property can be associated with either nodes or on the edges. And we also have edges that usually describe a relation. So this is really a simple and explicit way of describe relationships among the entities. And why we need a knowledge graph. The first reason is it's more intuitive to represent biomedical knowledge. Just think about the same knowledge graph I just described. This variant is a GBAS hit with T1D, and it's a eQTL of the gene CFTR, and the variant is also located in the gene body. This gene CFTR is expressed in pancreatic tissue, and this gene belongs to this ontology term. So we can use this network to describe their relations. But without this, I think traditionally people will use tabular data format to record the same information. We will have a table for GWAS hits. We have a table for eQTL signal, and we will also have a table for RNA-seq datasets. So this really describe the knowledge more intuitively. Secondly is in a research domain, like in type 1 diabetes research, there are usually tons of resources already there. And for example, for the same gene CFTR, they can be referred in example with this ID in PubMed, in this PubMed article. In GTEx, there's another identifier, and also in string, it has a different information, and in gene ontology, it has a geo term. So therefore, if we can put things together in a knowledge graph, we can really provide provenance. When we talk about the gene CFTR, we can go to all these resources so that we can summarize all the knowledge. So given what's going on in the type 1 diabetes research, we really need a tool like knowledge graph to handle this problem. Another reason is really it promote file principle. So when we implement the knowledge graph, we will use identifiers. So the node and edges must have unique identifier. Ideally, they can be a global unique identifier or eternal identifier that is widely used. And second, when we define the nodes, we also need to refer to a collection of schema or metadata. For example, the property need to be predefined. For example, here, the gene must have the strand information. It can only be positive or negative. And on the edges, this function annotation can only go from a gene to gene ontology term. So this all has to be predefined. And we also have a collection of ontology term to make sure we really use the uniform vocabulary. Just gave an example, research resource identifier RID is a unique identifier that is used in dkNET, and also will also use in our PanKgraph. That's really a lot of helpful to enhance the data integration, standardization, and also long-term accessibility. So this is something we are really happy with. Just next is about the scope of PanKgraph. Right now, we are just finished the first year, but right now we already have 500,000 nodes and 1.6 million edges, and it's still growing. It includes basic knowledge in type 1 diabetes and islet biology. We also have high quality data and knowledge contributed by the PanKgraph team and also the broader community. So right now we have the GWAS hits, the eQTLs, fine mapping and colocalization, and more are coming in future years. We also have more than 1,000 papers from HERN, and that is also being processed to enrich the knowledge graph. We also include a lot of annotations from other repositories like GTEx, Ontology, and dbSNP. And also within the PanKgraph, PanKgraph allow you to link to different resources. Like in PanKgraph, we have data library, we have analytical library, and also we can easily use PanKgraph to link to these external resources and also internal resources. So everyone, you are encouraged to go to pangraph.org to try it now, but I will not change my screen. So this is just a quick demo. So right now we can choose a question with SNP serve at the QTL for the gene. And then once you put the gene, the question will be updated for this particular gene. And then you can click search. And then that will promote you to this intermediate result page. Because in the PanKgraph, we have 4 types of QTLs already summarized. So they are listed under here. And for this particular gene, we only have 3 credible sets for splicing QTL. And you can click one of them, and that will give you to the final result page. So on this page, this is essentially for the question, how does this lead SNP influence the splicing of this gene as reported by GTEx? And we use AI answer to summarize what we know and also resources relevant to this answer. And also there's a part about you may also want to ask a different question. So behind the scene, the AI answer is really powered by large language models that can give an overview to the question. And then because we have this literature summarized in the knowledge graph, we can provide real literature references here like PMID. So this help ground the LLM and support explainable AI. And again, the resources will link you to the PanKgraph data library or analytical library and internal resources outside PanKgraph. And here we also have recommendation along the path on knowledge graph that may give you the next question you want to ask. Right now, PanKgraph also have an API implemented. This is really for people with computational background. So for example, if you want to identify all the variant linked to the gene in a particular pathway, you can just write this cipher and then execute and got 194 variant about 3 genes. So the difference between API and the web portal is web portal only allow you to perform one query, but the API allow you to subquery in a batch mode. Next, I would like to describe other 2 knowledge graph we built before PanKgraph. One is called genomic KB, one is called genomic literature KB. Both of them built a few years before PanKgraph. We really gained a lot of experience, and many of the functions is really served as a prototype for PanKgraph. They are different. So in terms of modality, the first knowledge graph, genomic KB, started 2021. It's really focused on genomic data integration. And then 2022, we started GLKB that act the PubMed literature. And you can see when we start PanKgraph, we just include not just data, literature, and also resources linked. And in terms of how to access the knowledge graph, so the earliest genomic KB right now we can do web query, API, we share the data dump. It can be accessed by LLM. And for GLKB, we started with query, API, and LLM, and machine learning. For PanKgraph, right now, we can only do web query and API. The LLM interface is ongoing. So the goal is to allow knowledge graph be accessed by different ways, either web portal or API or entire data dump or LLM or machine learning. So gradually we will enrich the utility of all these three knowledge graphs. So next, I will describe genomic KB, the first knowledge graph that motivated to integrate human genomic data. So this really, by the time 2021, there's already tons of genomic data available, like the ENCODE, 4DN, and GTAX, and Phantom5. All this, they describe different modality of human genome. And I noticed my collaborator always stare at a contact map along with genomic tracks like this. This biologists usually stare at one genomic region and compare the common contact pattern along with what they observe the peaks along the epigenomic tracks, and also eQTLs they identify from GTAGs. They try to identify the patterns among these, but you can see this is really difficult to kind of scale up. Even when you look at one region, if you are interested to see a similar region like this, you cannot just do this by eye by dragging the genomic browsers. So that's really the moment motivate us. Can we digitalize the information, the knowledge into nodes and edges, and then we can do more things systematically. So this is really the what we did. We integrated over 30 data sources, and we explicitly represent the knowledge at node and edges. So here is just one example. Here is type 2 diabetes. It has 3 GWAS variants like this. For the first one, is located on enhancer, and that regulate gene IL-10. The second variant is located in this epigenomic peak, and that form a loop with the genes TSS, DMRTA2. And the third variant correlated with the gene is eQTL of this gene TIMP4, and it's located in this gene, PPARG, and it's expressed in pancreas tissue. So this is really to convert knowledge in this genomic browser into a knowledge base, into a knowledge graph. So we have 300 million entities, 1.36 billion relation, and almost 4 billion properties. That took about 500 gigabytes on the space with the Neo4j implementation. So this is really put things, the data, convert data into node and edges to present the knowledge. And then what things became machine learning or computational ready format, we can ask a lot of questions. For example, people may ask the first question, which of the GWAS variant are located at the enhancer? And then you have a list of variant, enhancer, and then you can ask a follow-up question, do this enhancer form prompt loop with the genes? Okay, you can identify, narrow down a subset of the enhancers, and then you can ask which of these variants are also eQTL with the genes in the relevant tissue. So there's really a lot of questions, follow-up questions about the relations of these entities. Once we have the knowledge graph ready, we can easily query the knowledge graph and answer the question. For example, for the first question, which of the GWAS variants are located at the enhancer region? then we essentially ask which variant located in enhancer that is a GWAS with type 2 diabetes. And then the follow-up question, do this enhancer form chromatin loop with genes? Then you can add a condition, this enhancer form a loop with a gene. And then the third question is, which of the variant are also eQTL with these genes in the relevant tissue? Then you can add another condition, this variant is eQTL in this particular tissue like pancreas. And then you can just add this and you can query the knowledge graph. So this really gives you a way to answer these questions about relations easily. Think about you can spend hours of time to answer this question if you only have data in this format. But once we have everything in the knowledge graph, just one query search and you can have all the result and without any ambiguity. So this is really the motivation of the first knowledge graph, genomic KB, and we use the same structure in PanKgraph since T1D has a lot of genomic components. I think next I will talk about genomic literature knowledge base. We started 2022. We are also working on to extract knowledge for her literature to enrich PanKgraph. So next I will describe how we build GLKB. So here is one paper published recently from Dr. Cal Gorton's lab. So there's one sentence in the abstract, for example, beta cell specific gene include INS, blah, blah, blah. So we hope to extract the knowledge, like the nodes and edges, and map to the vocabulary using information knowledge extraction pipelines. So essentially here, we extracted the entity beta cell, the entity insulin gene, and then the article mentioned both entity, and we map the entity to the vocabulary to enhance, to make sure there's no ambiguity. So this is the entire pipeline. We first get a controlled vocabulary by this ontology resolution. And then we can extract the entity and then normalization. And the third step is evaluate the entity to make sure the entity extract are making sense. In the end, we will summarize the relations so that we can build this literature knowledge graph. You can see here, we have articles 1, 2, 3, entities 1, 2, I think 3 and 5 were dropped during the entity evaluation, 4 and 6, and then we have 4 vocabularies. So essentially, this is how the knowledge, literature knowledge graph look like. So this is the scope at this moment. We have almost 700 million nodes, 2.4 billion relations. It involves over 1 million terms, and it has multiple level of embeddings for abstract, for sentences and the terms. And we have web interface, API, and also embedding available. So how we can ground our large language model in question answering. So this is really the power of knowledge graph. When we answer biomedical questions, we can always refer to the real PubMed articles. For example, right now we are asking the question, how are gene RFX6 and PDX1 related to pancreatic development? So the planner will break this question into 2 subqueries and then identify 2 papers. And then there's the graph retriever. In the end, we can generate an answer and then also refer to these 2 articles by referring to the PubMed ID. So we test the pipeline on our tool on PubMed QA, and it outperforms the traditional method. So this is really the way using graph reg, retrieve, and use LLM to generate the answers. Also recently, we have been working with a biologist, Dr. Shuibing Chen from Cornell. So we are working on some project and because math trainees do not have a lot of biomedical training, and we ask a lot of questions like because Dr. Chen generate iPSC cells from patients, diabetes patients, and we don't know whether the iPSC are the same or different if the patients undergo a change in their disease condition. For example, if this patient take a drug, will the iPSC derived before and after drug the same or different? So at that time, we don't know the answer. So since we have the GLKB, we just asked the GLKB, which has the IOM agent. So iPSC in diabetic patient, will it change by disease condition, or is it like WGS not change in life? So this is just a question. And then our GLKB's LLM agent tell us will not change, and also giving a lot of explanation and also reference to the real articles. And we also tried this question with ChatGPT, and GPT gave us a really fancy answer, really well structured, but it says along this column, iPSC, the epigenetic influence can be altered by disease status and persist after reprogramming. So in this situation, GLKB and ChatGPT really gave different result, and we really don't know the answer. And we asked Dr. Chen, and Dr. Chen confirmed GLKB gave us the correct answer. So this is really a situation. ChatGPT gave a really fancy answer, seems really, really well structured, but actually it's wrong. And for computational biologists, without asking domain expert like Dr. Chen, we will never know the right answer. But I think GLKB does better because it always refers to the real article. Okay, so I have shown you the first is for a question, we can just do query and get the answer. And the second one is by using graph reg to retrieve the article and use LLM to generate a grounded answer. And we also ask, can we use deep reinforcement learning to propose new hypotheses? Like for the question, what is a possible mechanism between RFX6 and T2D? We are hoping we can train a deep reinforcement learner to learn new hypotheses. So this is an ongoing work. Is hypothesis generation use deep reinforcement learning. So the question is, what's the possible mechanism between RFX6 and type 2 diabetes? So we will have the agent traverse our knowledge graph, and the trajectories found by this agent will give some context. That is the sample, the scientific fact. And use that, we can generate hypothesis and justification. So this is really ongoing work. I think this is really one step even beyond question answering. We are generating new hypotheses. I think that's all for the talk. I would like to acknowledge in the first genomic KB is mainly the training in my lab. Once we move to a genomic literature knowledge base, we have more collaborators join the team. And of course, for the PanKgraph, we have a much larger, really well-organized large team supported by NIDDK. So it's amazing to work with a large group of people to build a knowledge base, knowledge graph like this. We would like to encourage everyone to try our genomic KB and fill in the survey, because we are right now trying to improve the portal. Any feedback will really be appreciated. And our genomic KB, the bioarchive paper is on as well. Without further ado, I would like to introduce my PhD student, Yuanhao Huang, who really the lead for genomic literature knowledge base.
Yeah. So, hi everyone. So here I'll give like a brief introduction to our web interface of genomic literature KB. So on this interface, we can use either graphical search or LM agents to retrieve literature evidence between different biomedical entities. Say, for example, I will try the graphical search first. So by doing graphical search, what we need is basically to type in multiple biomedical entities. Say, for example, we can use the example query here. We are typing in the SPRY2 gene, RFX6 genes, HNF4A genes, and type 2 diabetes. So, and then when clicking search, we are we're asking like what potential connections between each of them are there. So here, the returning results would be in the form of a subgraph. And each edge here represents a connection by the PubMed articles. Say, for example, we can click on one of these edges, and it will show that like the connection between the connection between RFX6 and HNF4A is like they co-occur in two of these PubMed articles. And say if you look at HNF4A and type 2 diabetes, like it tells you that their connection is association, and there will be like a brief summary that HNF is 30 in pancreatic beta cell function and insulin secretion. And along there will be some related sentence from the PubMed articles mentioning their association. And you can also click the view source. Yeah, so there's a bug here, but yeah, so we can look at the original source here. So, and besides the sentences, we also provide like the articles here, related articles that mention the association between the two biomedical entities. And so also we can also directly ask like, and using RAG methods, we can ask the large language model to generate to summarize the results from the provided graph and to like generate answer for any of these questions related to the graph. And also we are able to look at like the information of each biomedical concept here. Say, for example, HNF4A will have its ID, meaning that this is like a standardized concept from the ontology HGNC. And we'll have some of the top articles about the gene here. And also when we can also like convert this graph to like a article graph, and this will show some of the related articles for these medical entities. Say, for example, this article mentioned both type 2 diabetes and SPRY2. And if we click here, we'll show more details about it. And so here's like a graphical search of GLKB. And also we provide a preliminary version of large language model agent to query GLKB using natural language. Say, for example, we can ask like, what is the role of the gene BRCA1 in breast cancer? And it will try to parse the input query and translate into different query strategies and planning to ask the GLKB and to get back to you by in a structured result. Say, for example, here it will tell you like BRCA1 is a critical gene in the breast cancer, and it will summarize some key findings from the PubMed articles and provide references. And we can also like continue the conversation. Say, for example, asking what are other these genes related to breast cancer. Yeah, so it will automatically use Cypher to query GLKB and list like top about 100 genes that associated with breast cancer. And we are now like continuous to improving this large language model agents so that it can support like more complicated results, like comparing different groups of results, or even like adding graphical search result to generate a hypothesis or something. And
Thank
that
you.
will be all. Well,
I think
okay.
we are ready to take any questions.
Okay, great. So thank you, Dr. Jie Liu and Yuan Hao for a great presentation and demo. So now it's our Q&A session, and then we have about 20 minutes left. So feel free to discussion or join the discussion or unmute yourself to ask any question that you may have or you would like ask question how to use or talk about how to collaborate. Please just free to unmute. Yeah. I'll give people some time.
Oh, hi, very nice talk. One of the big problems that we've always had, having lived through all of the introduction of semantic web linked data, right? Basically the creation of knowledge bases, is to make them complete and keep them up to date. How have you addressed those issues, or will
This
you
is
address those
more
issues?
about sustainability, right? How to maintain this, right? So I think right now we downloaded the entire PubMed in 2023. Therefore the GLKB
Is that abstracts or?
I think right now we have all the abstract. We also have processed the full article, the PubMed Central. There's some work going on as well. Right now it's not in
Right.
GLKB, but we have ongoing work extracting the entire full.
But that's only a subset of PubMed Central, which is a subset of the literature, which is another problem. But anyway,
Exactly.
yeah.
The
So keeping it
pipeline,
up to date, I think, is really the important thing.
right. So the pipeline, I think, is semi-automatic. Yuan Hao, I don't know. I think it takes maybe weeks to reprocess everything. Yeah, I think that's kind of certainly there's LLM used during the entity linking, whatever. So if we have a powerful machine and you want to do a better job, it may be slower. But I think we are looking at weeks to update the whole literature knowledge base. Yeah.
And have you done a comparison? I mean, you said you're just getting the full text, but have you done a comparison of full text versus the impact of having the full text versus just the abstract on the information?
I don't think that's a great idea. We haven't done that. And the motivation of doing the full text is because full text has the figures. We hope to, in the future, when GLKB or PanKgraph generate an answer, it not just refer to the article, it can also pinpoint to the particular motivation value of article. I think understanding the full article is doable, and I think right now the large language model can do that. But I think our unique stress is really provenance, provide links providing evidence and grounding LLM. So that's why we are focusing on kind of extracting the figures. And also in PanKgraph, we are also hoping to extract the like RIDs. We can get resources or linked. So it's no longer data literate, but also resources, pipelines, anything we can imagine we want to consolidate. And I think another benefit is, I think this is really a discussion also in Pank-based team. I think there are two general mode. One is integrate. We can summarize and integrate everything into one centered database. There's also a way kind of linking provenance, linking to eternal resources. I think I think it's a trade-off. And I think right now we are also working and discussing. I feel both can be useful. I think for the most core resources we want to integrate, and then for the eternal that can be updated along with other resources, we probably want to link using knowledge graphs. And the ID is identified, like our ID is super helpful for us to do that.
Yeah, no, thank you. And I also think, whereas in the past, the one thing that we didn't have were the LLMs as a means to query the knowledge bases or the knowledge graphs, because they're very complicated oftentimes to query unless you know the structure. But the LLM now lets people just ask questions of them. So I think they're going to be even more powerful than they were.
Certainly, thank you.
Okay, any other question? You can also use the chat function. Yeah.
You know, thanks for the great talk. So one question about sort of the validation, right? You showed a couple examples, with the breast cancer gene, but that's pretty strong evidence, right, in the literature, and is a a very I would say solid claim. So I mean, what's your strategy for testing your various LLM responses, deep learning responses, to make sure that they are returning reliable results, right? Because researchers are going to be asking questions not about potentially the very strong already prior validated claims or hypotheses, but are going to be asking questions for things that may not have heavy support in the literature, or there might be conflicting support, right, in your knowledge graph in the literature.
Yeah, I think that's a great question, and also a long question. I feel right now we are still focusing on extracting. We have we have some results. I think I'm happy to share the screen again. I didn't get a chance. I didn't put these slides because the result we just got this week. We have been benchmarked our LLM agent with ChatGPT on a few PubMed QA answer benchmarking task. Most of them GeoKB agent works better and is evaluated by many criteria. I think probably the best way is having the biologist try and then provide case by case. I think that's probably more useful. I think numerically we can only get some sense, but like the example I just showed with shipping chain, the IPSC, this type of question probably is more useful. I think can be used widely by student. I just feel this might be a particular use case is more useful to tell us how well it performs.
Yeah, and I think with the slide you showed a couple sort of established benchmarks. So the sort of the follow-on question is could within for example the DK space, or because there are a number of activities in terms of AI applied to research areas in DK, like could we start collecting information about, potential benchmark questions, that are specific, right?
Right.
To
That's
related research. So,
yeah, that's great suggestion. I think early this week, the Punk-based outreach team discussed how to reach to machine learning and AI community. I feel for them, if we have a benchmark data set, maybe created by Marcella or Anna Goloin. So I think this domain expert can be a really powerful one. And right now, all these benchmark data sets like PubMed QA are really short, like 100, 200 questions. I think for in the DK space, if we can have the expert create a list of questions, I think that would be super helpful.
Thank you. Any other more questions from the audience?
yeah, I saw Suching
Okay,
on her video. So
yeah, Shuqing.
Thank you, Kowei. Thank you, Jay. Great talk. Very intriguing. I have maybe a naive question as a amateur of this topic. I was particularly fascinated with the large language model based agent. And it seems the field are developing a lot of AI agents for various purposes. People are developing for gene annotation, for various omics, etc. I was wondering, what do you think the future should be like? Whether we should be developing omnipotent AI agent, or should we develop like a fit for purpose AI agent and have them talk to each other and coordinate? I'm asking because they are large language model based, and the LLM being language based, they assume naturally they can communicate with each other easily, right? So I guess the question is, should we build bigger ones, or should we build specific ones and let them talk to each other and coordinate toward solving like grand challenges in biomedicine?
I think that's a great question, and I don't have a exact answer, but I feel either way, what we are working on for Punk-based, like building the data, consolidating the metadata, consolidate the identifier, integrate, and maybe have the provenance. This all the infrastructure made it either way possible. And I think, yeah, I cannot predict future, but I just feel what we are working on now are meaningful. And I feel both may work out, and maybe some may work better in some situation. The other one may work better in other situation. And I'm sure the research community will explore both, and also time will tell us which one. And maybe essentially both works well.
Maybe I can add a little bit about it.
Go ahead.
Yeah. So so actually we actually think about it quite a lot when establishing the GLKPLM agent. So right now we are using a multi-agent architecture. So say for example, we have a smaller agent called a planner to parse the user query into different subqueries and assign them to like different other agents. Say for example, when we ask like which gene is associated with breast cancer, it will parse this query and to like predict like which agent to use. Say for example, it says it might be a cipher query. It might be a query that better to answer using cipher query, and then it will assign to a cipher agent so that the cipher agent can translate this natural language query into a cipher query and query the background. knowledge base and summarize the return and summarize the answer and generate the answer. And so, for example, when we ask like specific question about the contents of different PubMed articles, and then the planner may use like the semantic search and use like semantic embedding to retrieve related articles. And by combining these different searching strategies, we are able to like answer more complicated question or even multi-document question to generate more comprehensive results.
Thank you. Thank you.
Thank you, Sujing. Are there any more questions, or is there anything that Dr. Liu want to add it to the presentation? No? Okay. If not, I think if anybody have more question, you can forward to me and then or send the email to Dr. Liu, and then I think he will be happy to answer. So thanks, Dr. Liu, and then also thanks Yuan Hao for today's talk. Thanks everybody joined today's webinar. A recorded version of this webinar will be uploaded to YouTube channel, and the slide will be uploaded to the SlideShare. So we encourage you to share all these resources with your colleagues and students. To receive upcoming webinar, please check our website or follow us on X channel, Facebook, or Blue Sky. So
There's one question that came in on the chat like a minute ago. So
okay.
Dave, you can see the chat, or?
Yes, I can see. So there's a question. I'm Ebenezer from the Department of Biochemistry, Osun State University, Nigeria. Would GLCK be useful for a low data set of SNPs from DNA sequencing? N equal to 2, say for each for control test 1 and test 2 for establishing correlation between two genes for disease condition 1, test 1 to another condition 2. It's yeah. So I think Dr. Liu probably can see the question in the chat, or if Ines, if you would like to unmute, please welcome to unmute. Yeah. Mm-hmm.
So I think the question is about whether GLKB is useful for a low data set of SNP from DNA sequences. Correlation. I'm not sure the question is about the GLKB or GKB, the genomic KB. Can you specify a little bit more? I suspect the question is more about genomic KB.
Can you hear Dr. J or Ebenezer?
Thank you very much. Good evening. This is
yeah.
Late Nigeria. We have a situation moment. We try to see in that subset. There are some of them that have CBDs, and when we did the PCR amplification, we didn't get amplification for all of the DNA. But because of lack of resources, we were just thinking, okay, how many can we find? So we have very low data set, and we are trying to see, would it still make sense? Ordinarily, logically, it should not. But I'm now looking at what we discussed about this GLCK, if it can go back to
I think
database.
I think I got.
Can it be of any help in any way?
So I think you have situation like the data is not that inclusive, but you want to use genomic literature knowledge base to explore and explore hypotheses, right?
Yes, please. Yes.
Yeah, I think in this situation, I think if you look at our manuscript, we indeed has one use case kind of for type 2 diabetes, exploring the gene relations between RFX6 and other genes in the context of T2D. I think the best way is to perform using the APIs. So so that you can systematically compare and perform significant tests. So Yuanhao, I believe in your paper, you already shared the pipeline for this purpose, right? And also in figure 6 or figure 5, I cannot remember exactly.
I joined quite late. If you help me with the link, I think I will appreciate that, please.
Right.
And the slides and the full presentation will also be made available.
Oh, great.
So at the end
Thank
of
you.
the slides, there's a QR code for GLKB manuscript. In that manuscript, we have our pipeline shared. I think you can try that pipeline, and you can reach out to me and Yuanhao if you have questions.